After discussing the different techniques for the DNA sequencing, briefly we can focus on sequence accuracy, that how much fidelity of sequencing all of these techniques may have. Although there is a lot of development in different sequencing procedures or technologies, but still there are most of the protocols that can introduce different degrees of errors during the DNA sequencing procedure. High performance automated sequencing machines, although have minimized the errors of DNA sequencing that are introduced due to human activities, but still problems are there. For example, in case of gel compression, still these errors are unavoidable. And most of the errors that are introduced by such problems are related to base substitution. In addition to base substitutions, some problems related to DNA addition or deletions are also there. And in molecular biology, such problems, they are commonly called as the indels. और in addition से या substitution से, चाहे वो single base की substitution हो या more than one base की substitution हो, उसकी वजह से जो reading frame है DNA sequence का, वो shift हो जाता है. और इस तरह से हमने जो प्रोटीन का सीक्वेंस या अमीनो एसिड का सीक्वेंस हमने प्रिडिक्ट किया होता है, वो इन्फ्लुएंस होता है और इट इज इफेक्टेड. एंड इवन द कंप्लीट सीक्वेंस ऑफ द DNA segment or even the chromosome, it is also affected. Now there is also a lot of development in the computational tool where we can minimize such errors, and especially errors related to frame shift mutations. They can also be detected by comparing all the 6 reading frames by using online databases. Here I can explain 2 examples related to the sequencing accuracy. And the first of the work, it is conducted in early 1990s, like 1992, by the group of Wilson and co-workers. They analyze a stretch of mouse DNA that is having the size around 96 kilobase pair. And they compare, after cloning in a vector, around 288 DNA sequences related to this particular DNA stretch. And they found that if the DNA stretch, it is beyond 400 base pair, then the frequency of error, it may be around 3.2%. And if shorter DNA fragment analyzed, like less than 400 base pair, then the sequencing error, it is 2.8%. And most of those errors were related to the ambiguities in the reading of the sequences. For example, at the end of the sequences, there were errors where 1 or 2 bases, they are repeated many times, and other ambiguities or errors were related to the secondary structure of the DNA. However, they analyzed the whole sequence of that DNA fragment, around 5.9-fold redundancy, and then they finally calculated that after analyzing the whole sequences, that the error frequency, it is now minimized around 0.1%. इसके अलावा एक और एग्जांपल मैं कोट कर सकता हूँ जो कि रिलेट करती है यीस्ट या स्कैरोमाइसिस के DNA की सीक्वेंसेस से मुतल्लिक है कि by using high quality procedures, 35 European laboratories, they analyze the sequence of a yeast, especially the 11th chromosome, and they find out that here the sequencing accuracy, it is 99.97%. that is more or less similar the examples that I quoted above. Now what type of quality procedures they adopted, I can explain here with the help of one table. This table, it related to quality control of sequence data from 35 laboratories that were engaged in sequencing yeast chromosome, for example 11, and different methods they used for verification. For example, in case of first one, original overlap between cosmets, where the cosmet they were used as a vector. The total number of fragments they analyzed around 28, and around 63,000 bases they verified, and they detected error around 0.02%. And then they again verified the data like resequencing of selected fragments that is having the length 3 to 5 kilobase long, and then resequencing was also carried out for random segments that is having the length around 300 base pair, and then resequencing of suspected fragments was also carried out. So in this way the average error rate was found around 0.03%. So by using different advanced sequencing techniques or technologies, we have sequence information, that is whole sequencing of different type of organisms. For example, different type of viruses have been fully sequenced. Number of prokaryotic microbes like bacteria, they are also sequenced. In eukaryotes, yeast, it is the most common example. In plants, Arabidopsis sequence, it is now available. And finally, the human genome sequence, it is also well famous, that has been fully sequenced.